you need to do.
Yes. Good. Go ahead.
Okay, so let's carry on. So yes, thank you again for this wonderful invitation. It's just always a buzz to meet new people that I have a lot in common with. So I've decided to organize my talk today around what I think is one of the most fascinating, scientifically important, and deep questions that one could possibly hope to devote one's scientific career to, which is, if I could just figure out how to actually, here we go, which is, how does a cell, which is the basic, the most fundamental unit of life, how does a cell think? Which is to say, how does this tiny living entity, the smallest entity actually that can be said definitively to be alive, how does this little entity represent the world to itself and make decisions and even learn based on its interactions with its environment? So as you know, inside each living cell is this dense profusion of interacting molecules, mostly proteins. And these form networks of interlinked chemical reactions. So these chemical reaction networks act in place of a central nervous system for the cell and act as a kind of a brain for the cell. And what's fascinating is that these networks have completely self-organized under evolutionary pressures and are completely self-regulating. So we're very interested, my students and I and our collaborators, we're very interested in understanding the network principles that are responsible for the extraordinary cognitive sophistication that is realized at the level of each living cell. And of course, this is a frontier scientific question, and being able to answer these questions will open up fantastic new avenues, transformative new avenues in biotechnology, therapeutic development, and so forth. So let's think a little bit about what these networks have to do in order to have this cognitive sophistication. And it's immediately clear that these network responses to incoming signals cannot be completely arbitrary. They have to be able to do very special things with the processing of extracellular or even intracellular signals. So I've chosen here two particular functionalities that are essentially the workhorses of cellular cognition. And these are by no means the only functionalities that cells rely on. Of course, there are many other things, oscillations and various other things, but they're quite limited things that cells actually have to be able to do in order to make sense, to extract meaning from biochemical information. So I've cherry-picked these two because I think these two are the most interesting and best understood. So on the left, we have switches, and I've chosen one particular type of switch, which is a one-way switch. There are many types of switches, actually: discontinuous switches, continuous switches, one-way switches, two-way switches. I've just illustrated a one-way switch here for illustration. And on the right, we have another class of response, which is an adaptation response or a desensitization response. So let me talk about just briefly about these before we move on, just to sort of get our minds in the zone about what kind of responses are important to cells. So on the left, I've got here a depiction of a one-way switch. This is actually the solution from a model I constructed years ago of the mTOR pathway, actually, in a cancer model. And what we're looking at here is actually the steady states of one particular protein in that pathway, which happens to be insulin receptor substrate 1, which is phosphorylated or activated at a particular tyrosine residue. So what you can see here is that for a particular threshold value of, let's say, an extracellular stimulus, S star, so this could be some growth factor outside the cell, like insulin-like growth factor, for example. So for concentrations of this growth factor that are less than S star, you can see that there are 3 steady states possible for this particular protein. There are 2 stable steady states, and in between this we have an unstable steady state. So you can imagine if we start with very low levels of this extracellular ligand, the cell, or this protein and things that are regulated by this protein will remain in this very low basal activation state. But if we increase the concentration gradually, at a certain point, at this threshold switching point, beyond this, only one steady state is now possible. So the system is propelled, therefore, to this high level of activation. And what's important about this function is that even if the stimulus is now completely removed, the networks, the proteins, the activation states remain in this very high level of activation, even if there's no stimulus anymore. And these kind of functions are very important in cells. They occur in many different contexts. A particularly well-known one is in apoptosis, for example. So in apoptosis, we don't want cells undergoing apoptosis just arbitrarily for no particular reason. We want this to occur only when the cell has received a decisive signal to commit this self-suicide program. But once it does so, we want it to absolutely commit irreversibly to this program. Because apoptosis involves the cleavage of intracellular molecules, including DNA, we don't want the cell changing its mind midstream and recovering from this process once its genome has been corrupted. It must be a one-way process. So this is a very important way for any cell to convert a transient response to a sustained long-term response. And again, there are many uses for this. We think this is also very important in cancer as well. We think that cancers can hijack this kind of response aberrantly, and this can give rise to constitutive activation of, for example, mitogenic proliferative signals, even when the cell is not being instructed to do so. And the presence of these kinds of responses have a lot of interesting consequences for therapeutic strategies and so forth, which we have studied quite significantly. So that's one-way switches as a particular example. So the example on the right is robust perfect adaptation, or RPA. This is actually quite a broad class of responses. It sometimes goes under the name concentration robustness, or desensitization, or homeostasis. But perfect adaptation basically refers to the independence of a steady state to the concentration of various stimuli to which the network is subjected. So if we have perfect adaptation, then the steady state is completely independent of these variations. If the steady state is weakly dependent on the level of these stimuli, we might call it imperfect adaptation. But robust perfect adaptation means that it has to do this without any requirement for fine-tuning of parameters. In other words, it has to be a network property, a system property, not a property of special parameter choices. And that's very important in biology, because biological systems characteristically cannot tightly control their parameters. It has to be able to implement these various functionalities under wide ranges of parameters. So that's very important. So I thought I might tell you a little bit about the history of RPA. It's very interesting. People have been interested in this for a long time. I should just mention, just for later reference, we call this baseline level that's in the steady state that's independent of stimuli, the set point. And this response is really a major workhorse of cellular cognition, because it gives the cell a reference point to compare incoming stimuli. It can detect if stimuli are high or low, or increasing or decreasing, by having this reference level. And this has been observed ubiquitously throughout all of biology, not just at the cellular level, but in biological systems more generally. It's everywhere. RPA is everywhere. So I've picked a couple of examples just to highlight this. You can see here, for example, in olfaction and our sense of smell. This is a very common example from our everyday life. We've all had the experience of being able to smell a fragrance of some type, but shortly afterwards we realize we can't smell it anymore, even though the fragrance is still present. So this is a very important feature built into biological systems that have this sensory function. Another example is chemotaxis in single-celled organisms. As I'll mention in a moment, chemotaxis in E. coli specifically is the locus classicus of the study of RPA in biological systems. We'll talk about this in a moment, but there are many other examples, including at the whole organism level. Many different organismal properties are subjected to various types of RPA and homeostasis, as we know, and we see this property ubiquitously in all kinds of intracellular networks, signal transduction networks, metabolic networks, and so forth. So again, as I started to mention a moment ago, let me just tell you a little bit about the history of RPA, because it turns out, even though it is ubiquitously observed and fundamental to what a cell has to do, it's actually not so easy to construct networks with this property. I mean, it's easier now that we know so much more about RPA mathematically, but naively, if we just start to construct networks that we think might have the property, it's actually quite hard to do this with this robust property, robust perfect adaptation. This is a very heavily curated historical viewpoint of RPA. As I mentioned, people have been interested in this for a very long time because it's been so widely observed, and we know how important it is. But the locus classicus, again, of RPA in biology from a mathematical point of view goes back to this landmark study by Barkay and Liebler that I'm sure you're all aware of. So in this particular system, I'm sure you're familiar, the property that's subjected to RPA is actually the tumbling frequency of the organism as it moves. So this is how chemotaxis works in E. coli. There are basically two types of motility in this organism. There's tumbling and there's runs, there's forward runs. So under normal circumstances, if there's a chemoattractant of some kind present at a constant level, then this tumbling frequency will be a constant, as you can see here in this diagram. And so the cell will essentially engage in a random walk. But once you add an increased concentration of this chemoattractant, this will stimulate the cell transiently to engage in this forward run. And this allows the cell to bias its motion in the direction of increasing concentration. So this is how chemotaxis in this organism works, and it's completely dependent on establishing these baseline reference levels of tumbling frequency under a constant chemoattractant or chemorepellent input. And this is a very simple, what's nice about this particular signaling system is that the chemotaxis response is due to a very simple signaling network, a two-component signaling network. So Barkay and Liebler were able to use this very simple network to construct a model that exhibits this constant return to a consistent baseline or set point in a way that does not depend upon special choices of parameters. Before this time, it was not clear really if in biological systems it was necessary to actually have special conditions on parameters to achieve behaviors like this. But this model was the first really proof of concept that this could be a network property, and they developed a simple model to show how this worked. Not too long after this, the same group, this time led by Uri Alon, did another very interesting study on the same model organism to show that actually you can change the set point by introducing various mutations into the organism. So this changes this constant baseline of tumbling frequency. And what was interesting about that is that that left the phenotype, the chemotactic phenotype, completely intact. So in other words, it's not the level of the baseline that matters for the phenotype, for this ability of the cell to interpret changes in concentration of a chemoattractant and undergo chemotaxis. It's the fact that it has a baseline. It doesn't matter what the value is. So this is very important actually in cellular cognition. Very frequently, RPA-capable systems do not depend on a specific value of set point. What matters is that it has a set point that it can use for comparison of incoming signals. So that was a very important discovery as well. So one of the next most important discoveries in the RPA field was this very well-known paper that I'm sure you all know, which is this paper by Ma in 2009 out of Wendell Lim's lab. So before this particular paper, people had thought about RPA from a mathematical viewpoint in very ad hoc ways, by one model at a time, constructing, by ad hoc means, ways to implement RPA. But this paper was the first paper to think more about what are the general principles that implement RPA. And so this was a computational approach, and by nature of being a computational approach, they were restricted to quite small networks. And in this study, they were restricted actually to 3-node networks, where they looked at 3 interacting molecules under various assumptions like Michaelis-Menten kinetics and so forth. And they considered all these various possible configurations of these 3-node networks, and they simulated all of these under very large parameter regimes to find which network solutions were able to exhibit RPA. So it was a huge computational task. And what came out of this study was that, at least for 3-node networks, there were really only 2 fundamental ways that such networks can implement RPA. That there are variations on these types, but they're all of these 2 very distinctive types. One is a feedback structured network, where very wide ranges of parameters were possible, but there were very strict constraints on the node or the protein that was embedded in the feedback loop itself. So they call that a buffering node. And there were very strict constraints under the Michaelis-Menten formalism on parameters regulating that node. Otherwise, other parameters were virtually able to be changed freely without disrupting RPA. So that was one particular solution. And the other solution was an incoherent feed-forward solution. So here again, very wide ranges of parameters could support this particular construction, but there were very tight constraints under Michaelis-Menten formalism on the regulation of this node that's embedded into one of these parallel pathways. So this was a very important study in this field because it started to suggest that the network possibilities for achieving RPA might actually be quite rare in the space of all possible network configurations. But of course, it also opened up a lot of new questions. So this really only applied for 3-node networks. It wasn't clear from this study what might happen if we start to consider larger and larger networks. If we consider 4-node networks, 5-node networks, would we find network solutions that are different in some fundamental way from these solutions that we find for 3-node networks? It's not clear what would be the situation once we open our view of study to larger networks. And we really can't study this in generality in a computational fashion. It simply becomes too computationally expensive very quickly. So even 4 or 5 networks, 4 or 5 node networks, because these networks grow in complexity factorially, we get too many configurations very quickly, and it quickly eludes our ability to study networks in this way. So I'll come back to this point about the more general case in a moment. Before doing that, I just want to touch on a couple more important studies in this area, just to sort of highlight the various approaches that have been taken to study this problem. So not long after the Ma study, there was a very influential study, which I'm also sure that you'll be familiar with, by Shinar and Feinberg. And they studied RPA in a particular form that they called absolute concentration robustness. This is a type of RPA where, rather than considering an exogenous signal, the disturbance that these signals undergo is usually the total expression levels of the interacting protein. So this sort of absolute concentration robustness form of RPA is often restricted to cases where we have a closed system, yes, that is isolated from exogenous inputs, usually, although we can extend this in many ways. So the method that they used and that has been extended since this time is largely based on an algebraic invariant known as deficiency. So that I can refer to this again a little bit later, let me just briefly elaborate on what deficiency is. I'm sure you know what this is, but let me, just in case someone does not know, let me just briefly.
Please go ahead, yeah.
Yeah, sure. So deficiency is a very easy invariant to compute in any chemical reaction network. It's a graphical, an algebraic property, basically, and it has three contributing components. One is the number of complexes in the chemical reaction network graph, which is simply the number of vertices in the graph. So in chemical reaction network theory, we call the vertices of the graph complexes. And these are, of course, multi-sets of species that interact to form the network. The second component is the number of linkage classes. And these are simply the number of connected components of the graph. And finally, we have the rank of the chemical reaction network. So this refers to the number of linearly independent reaction vectors in the network. So just for illustrative purposes, I've highlighted here the three reaction vectors that correspond to the first linkage class. So for example, if we order the species A, B, C, D, E, F in order here, the first reaction converts two molecules of A to B. So in the first reaction vector, we have a -2 in the A location and a +1 in the B location. So we can do that for all the reactions in a network. And these reaction vectors together span a vector subspace called the stoichiometric subspace. And the dimension of that subspace is the rank of the chemical reaction network. So all of these quantities are very easy to compute. The first two are simply a matter of counting, and the rank is simply a simple linear calculation. So it's very easy to determine the deficiency of a chemical reaction network, even for very large networks. And I'll mention just for future reference that complexes it's helpful to partition the complexes of a chemical reaction network into non-terminal complexes, which I've highlighted in blue, and terminal complexes, which I've highlighted in pink. And these mean exactly what you think they would mean. And I've illustrated these here. Shinar and Feinberg did a very interesting study using deficiency, and they proved a very important theorem, which has had a huge impact on our field. And I should just actually go back and just highlight the fact that this particular chemical reaction network that I've highlighted just for illustrative purposes has a deficiency of 1, and that is because it has 8 complexes, 2 linkage classes, and a rank of 5. It's very easy to make that computation. So 8 minus 2 minus 5 is 1. This chemical reaction network has a deficiency of 1. And there are many theorems actually that apply to chemical reaction networks that have deficiency of 0 or 1. And the Shinar-Feinberg theorem is an example of a theorem that applies to mass action chemical reaction networks with a deficiency of 1 specifically. So the theorem states that if these reactions proceed under mass action kinetics and the deficiency of the network is 1, then if there are 2 non-terminal complexes that differ only in one particular species, then the system has RPA, or what they called absolute concentration robustness, in that particular species. So again, this is happens to be another very simple example of a two-component signaling system in E. coli, which has a deficiency of 1. Actually, it's very easy to compute this. And you can see here that this particular chemical reaction network has 2 complexes, XT, which is a membrane protein, and XT plus YP, which is another non-terminal complex. And because these differ in YP alone, then we are guaranteed that since this is the deficiency 1 network, this system will exhibit RPA in this particular molecule, YP. And what's important about this result is not so much its practical utility in and of itself, because the truth is very few chemical reaction networks in nature have such low deficiency. Most chemical reaction networks that are really of interest to us have much larger deficiency, and any network that has deficiency greater than 1 immediately eludes this theorem. So it's not so much the practical utility of this theorem in and of itself that's important. What's important is that this is one of the rare examples in biology where we have an actual theorem that tells us something that's definitively true once the conditions of that theorem are met. So as you know, mathematical biology is dominated by modeling, construction of models, and analysis of models. But a theorem is very different from a model. A theorem, again, tells us something very definite about systems that have the properties that are captured by the hypotheses of the theorem. And this way of thinking has really driven research in this area forward tremendously because of this way of thinking. And I'll tell you more about where some research at least has gone since the Scheiner-Feinberg theorem arose over 10 years ago. Let me just briefly tell you one more thing. I might just skip over this quite quickly, actually, because I think I'm going to run a bit tight on time. But I did want to mention just briefly that although perfect adaptation in biology has a relatively recent history of only 25 years or so, this property actually goes back much longer in the context of engineering control theory and goes under the name robust asymptotic tracking. And this property corresponds to what control engineers call integral control, feedback integral control. And there have been many interesting examples that have been able to relate this control theory principle to RPA in signaling networks. And I've highlighted this one quite important example here. I'll just gloss over this quite quickly so that I don't spend too much time on it. But this is an example that's called antithetic integral control. And just quickly, I'll gloss over the fact that these particular interactions are captured by these very simple mass action equations. And these interactions are embedded in a feedback loop. And you can see here that a very simple linear coordinate change obtains from these chemical reactions what we nowadays would call an RPA polynomial. So this is so when we integrate this transformed equation, we obtain an integral variable. And this integral actually corresponds to the integral of the error between this RPA capable variable XL in the network and its set point. So this is how control engineers typically think about robust asymptotic tracking, which is equivalent to RPA. And the fact that this integral, let's say, can be obtained through a linear coordinate change turns out to be very nice and very useful and something that we can think about in a more general way later. So I'll come back to this point in a moment. But this leads us to a much bigger question regarding how can we think about RPA in the context of arbitrary cellular complexity. A lot of the work in this field has focused, as I've mentioned, on either ad hoc approaches or computational screening of small networks. But how do we understand very large networks? What are the general principles that capture, that are able to implement RPA in the most general possible setting? Because the truth is, particularly in the context of mammalian signal transduction, we might never really know in complete generality all the tiny intricacies of these highly, highly complex signaling networks. So what we really want to do is work backwards. We want to answer the question, what are the absolutely general principles to which all networks, regardless of how large or how complex they are, what are the general principles that all networks are subject to in order to implement this keystone functionality? And so we've done a lot of work on this over the last 5 years in particular, and I'm just going to tell you about this really briefly. So the first paper that I'm going to tell you a little bit about on the left is concerned with the macroscale RPA problem, concerned with the general network design principles that can implement RPA in networks of arbitrary size and complexity. In our more recent work, we've shown how these very general principles can be realized at the network microscale, at the level of detailed intermolecular interactions. So let me just tell you a little bit about this briefly. So first of all, let's talk about the general principles of RPA in the context of complex networks. So as I mentioned earlier, we can't think about this from the point of view of concrete networks and computational screening. We have to have a more abstract way to think about encoding biological complexity and the constraints to which RPA capable networks are subject. So the question is, if we allow ourselves to think about networks that are bigger than 3 nodes, at what point will we have solutions that are distinct from the three node networks that Ma discovered? And are there an infinite number of distinct solutions, or are there really a finite number? Are these finitely generated? Or might it be infinitely generated? Is it like the prime numbers? You just keep getting, as you get bigger and bigger, you just find more and more solutions. And what we discovered actually is that there are no new solutions as we get larger and larger network sizes. All networks that exhibit RPA do so through a decomposition into basis modules. And these basis modules are potentially quite complex generalizations of the simple solutions discovered by Ma. So let me just tell you a little bit about these generalizations of the 2 solutions. So here on the right, I'm talking about the feedback structured network. In the general solution, we call these opposer modules because they're more complex, generally speaking, than the 3-node solutions that Ma discovered, which I'm showing here on the right of this panel. So in this very abstract framework, we're able to discover very general properties of these feedback structured networks. But they all have this, what unifies these solutions is this essential feedback structure. And so embedded into the feedback loop could be quite complex networks of interlinked feedback loops, actually. And interspersed in these feedback loops are special nodes called opposer nodes. And these are generalizations of the buffer node that was identified in the Ma study. The Ma study was restricted to Michaelis-Menten kinetics, whereas ours is more general. We have a very general way of thinking about the constraints on the regulation of these particular nodes. So we know how these nodes must be regulated, and we know how these nodes have to be distributed in these feedback loops. So I'll tell you a little bit about our method in a moment, but essentially the idea is to induce network structures relative to a chosen input-output pair. So what I mean by that is any network that exhibits RPA has to exhibit RPA in response to at least one exogenous input. So there must be at least one component of the network that receives this input. We call that the input node. And it must exhibit RPA at least one node, which we call the output node. But once we've induced a structure relative to that input-output node pair, we can find the very general properties of these network structures. And in fact, usually these networks will exhibit RPA at nodes other than the chosen dedicated output node. And we can understand these completely where within these network structures, where embedded nodes exhibit RPA. And I've tried to depict this schematically by attaching these little red asterisks to nodes within the network that also exhibit RPA in addition to the output node within these induced structures. In addition, these networks can generally exhibit RPA when stimulated at other nodes in the network beyond just the originally identified input node. So this really generalizes to a multi-input, multi-output a network situation once we've induced these network structures for a particular input-output pairing. So anyway, this is the solution for the opposer module, which is a generalization of the feedback structured motif that Ma discovered. And the other module is a generalization of the feed-forward structured module discovered by Ma. And I should point out that on both of these module types, this E symbol that I've highlighted here means that we can actually embed arbitrary subnetwork structures into these locations without affecting RPA, as long as the network remains stable. So again, this is a generalization of this feed-forward structured solution. In general, these modules can have arbitrary numbers of feed-forward pathways, and all the nodes embedded into these feed-forward pathways are subjected to very special constraints that we call balancer kinetics, and we understand everything about how these balancer kinetics work. And the node which joins these parallel pathways together is called a connector node, and this is also subject to some special kinetics. But in any case, these are the general features of these modules that are a basis, a topological basis for all RPA-capable networks, and these can be connected together to form arbitrarily complex RPA-capable networks. So I was planning to tell you a little bit about how this solution strategy works, how we came up with this solution. Maybe I'll just go through this very quickly in the interest of time. I'm happy to talk more about this maybe in the Q&A later. But essentially, the basic idea is to consider the structure of the derivative of this system. And we can easily come up with an algebraic constraint that represents the presence of RPA for a chosen input-output node pairing that we choose in the initial instance for the purposes of deducing these induced structures. So this is a potentially quite a large equation in generality, because it can contain up to n-1 factorial terms for an n-node network. So it could potentially, in fact, it will contain n-1 factorial terms for a fully connected network. It'll contain less than that if it's not fully connected. So this is a very large equation to study in generality. And the way we study this in generality in the abstract is to actually not consider this as a sum, as we would normally do with a determinant equation, but we actually consider this to be a collection of elements that carry a sign with them. The sign is inherited from the determinant structure, and is also influenced by whether these various interactions are activating or inhibitory in nature. So this gives us a set of terms that carry a sign structure with them. And our general approach is to actually partition these terms into subsets which sum to zero independently of all other subsets, bearing in mind that these the terms that form these sets contain common elements in the term of partial derivatives, and cannot give rise to constraints that contradict the constraints for other subsets. So it's actually a very complicated problem, which we've now solved in complete generality. But there are basically two types of sets that are induced in this partition. Some sets achieve this zero-sum condition by having each term in the set to be identically zero for all perturbations to the network. And these correspond ultimately to the opposer modules. Other types of sets are strictly non-zero in nature, and these sum to zero for all inputs, and these correspond ultimately to the balancer modules that I just mentioned. But this, and I should just mention briefly, I was just going to provide a bit of an illustration. I've just shown you here, for illustrative purposes, here are some extremely simple network constructions that are capable of RPA. And I've chosen these because even though they do contain quite a few nodes, they're sparse enough that they only contain three terms in this particular algebraic condition. So it's very easy to see for these simple cases how these achieve RPA. And again, just to emphasize, we can understand in a very definitive and concrete way, once we have an induced structure, we're assured that certain nodes, in addition to the dedicated output node, also exhibit RPA, as I've indicated with these red dots. So we can really understand RPA in a very general way using this theory. And just here are a few very simple examples, just to show how slightly larger networks conform to this theory. So what we'd also really like to do is understand how this would work at the detailed intricate molecular level. So for example, if we go back to the Shinar-Feinberg example of deficiency 1 networks, for instance, it would be nice to be able to reconcile chemical reaction networks that exhibit some form of RPA, like absolute concentration robustness, with this very general theory to understand in a very general way how chemical reaction networks can exhibit these this special property. So let me just return to Shinar and Feinberg's example, just to talk a little bit about how we've generalized that work using our prior methodology and using a generalization on how deficiency affects algebraic invariance in chemical reaction networks. So let's return to this very simple example, which happens to be an osmoregulation two-component system, which Shinar and Feinberg considered in their paper. So this is a deficiency 1 network. And I've just used neutral variables here, x1 through x8, just to make this a little bit simpler to talk about the mathematical principles. So the molecule that actually exhibits RPA or ACR in this case is this one here, phospho-MR, which I'm calling x2. So we know from the Shinar-Feinberg theorem, because this is deficiency 1, we know that under the mass action framework, the monomial associated with this particular non-terminal complex, x1, x2, and the monomial associated with this particular non-terminal complex, x1, will be, there'll be a constraint on those monomials as given by this particular polynomial. So this polynomial is guaranteed to vanish at all steady states of this particular system. That's guaranteed by the Shinar-Feinberg theorem. And this particular parameter here, B2, will be a function of the parameters of the network. In fact, this is true for a polynomial of this form will hold for any two non-terminal complexes. So for later use, I've also noted here this particular polynomial, which relates these two monomials here, x1 and x3, which are also associated with non-terminal complexes in this deficiency 1 network. This is not particularly relevant to RPA in this particular network, but it will be relevant in a moment when we consider a more detailed network. So this is actually a polynomial that we would call an RPA polynomial. And you can see very clearly its relationship to RPA. So this polynomial vanishes on all steady states of the system. So if you can see, if we factor out an x1, we can see that x2 is constrained to take the value b2, which is a function of parameters at all steady states of the system. Mm-hmm. But what happens when we consider more complicated versions of this chemical reaction network that don't have deficiency 1? Mm-hmm. So Shinar and Feinberg did actually think about this problem, mm-hmm. And they mentioned this particular network in their paper. But this network eludes their theorem because we've added this extra linkage class here, which increases the deficiency by 1. So this is the deficiency 2 network. So Shinar-Feinberg theorem says nothing about this particular mm-hmm. network, even though it's very easy to show actually mm-hmm. That this network still exhibits ACR or RPA okay. At x2. And so what we've been able to do actually is to be able to show how deficiency controls the structure of algebraic invariance in a network. And this turns out to be quite complicated, but we've been able to do this. And I just want to highlight the fact that we can show actually that even for this deficiency 1 case, not only does this polynomial, both of these polynomials actually, not only do they vanish at all steady states of the system, but these particular polynomials mm-hmm. are always, for sure, obtainable through a linear coordinate change of the system's chemical rate equations. Mm-hmm. And that is guaranteed to be true. Mm-hmm. And that's very useful. And so in the more general case, for larger deficiency, we can show how these linear algebraic invariants evolve with evolving deficiency. And so in this particular case, the consequence of adding this extra linkage class is to actually add this particular monomial, which is associated to this non-terminal complex, to this invariant, which is in the row span of the system. That is to say, it's obtained by a linear coordinate change on the reaction equations. And we're able to relate this to the space of all possible RPA-capable chemical reaction networks by noting actually that these are very special row span polynomials. And we call these, in this particular case, the first is a balancer polynomial, and the second is a connector polynomial. And that's true because we're able to actually relate this particular chemical reaction network structure to a balancer module that I mentioned earlier. So these are the building blocks of RPA for this particular network. There are always building blocks of this type for balancer modules. And we can combine these linear invariants to obtain, once again, an RPA polynomial. And we can do that in a very structured and principled way and show how RPA obtains in this network. And again, this is a consequence of the fact that topologically, this turns out to be a balancer module. And all RPA-capable chemical reaction networks can be related to these modular structures and have these linear invariants that relate in very special ways to these modular structures. So this particular network is a balancer module. We've looked at many other examples that have the opposer module structure. I'll just gloss over this. I think this is just relating these these invariants to impose the RPA on these subsidiary parts of the network that relate to the topological structure of the module. But this is another example of a chemical reaction network that exhibits RPA. This happens to have a deficiency of 3, and that turns out to be because this is a 2-node opposing set, which we can relate to our study of opposer modules. And again, we can obtain these invariants that are strictly in the row span of this chemical reaction network. So by relating the development, the evolution of algebraic invariants in the row span as controlled by deficiency, we can actually relate chemical reaction networks in this very detailed way to macroscale topological features of the network, thereby obtaining a very general description of chemical reaction networks that can exhibit RPA. So once we have this very general way of encoding RPA, essentially implicitly, we can use these principles to define the space of all possible chemical reaction structures that have RPA. And we can use this now to think about evolutionary processes. As new reactions are added to existing networks, we can understand in a very principled way how networks may preserve the capacity for RPA or lose the capacity for RPA as these particular network solutions traverse network space over evolutionary time. This has a lot of biotechnology consequences, therapeutic consequences. And so it's very interesting, and we're very actively working on many of these corollaries in our ongoing research. So that we have enough time to talk together, I thought I'd just wrap it up there and just wanted to thank my generous supporters and funding, and of course I work with many wonderful students here at QUT, and I look forward to working with many wonderful students in the future on these problems as well. So thank you so much for having me, and I look forward to talking to you both now and hopefully in the future in person as well.
Yeah.
Thank you.
Thank you, Robyn. Okay, so let me